Hello, everyone, and welcome to our session today, which is about general characteristics of the CNS, central nervous system, and the PNS, peripheral nervous system. Okay, let's dive into it. It's a very nice topic and quite a short one, actually. So let's dive into it. First, you guys need to know some general info, some typical things you need to know before we go deep into what's the difference between the two. So you need to know that both the CNS and PNS are part of the nervous system, so their origin is almost the same, which is from neural ectoderm. And the important cells in the nervous system are two cells. There are neurons and there are glial cells or supporting cells. And next, you need to know what's a general definition of nervous system. Nervous system is a network of communication between neurons which are communicating via neurotransmitters to allow the signals to pass through them by action potential. Now, there are two types of signals we can have. We can have afferent signals and efferent signals. What's the difference? Afferent, it's sensory, meaning that it goes from the receptors, for example, at your skin. The receptor will feel something. I don't know, it could be maybe a burn or something like that. And they will send this info all the way to your central nervous system. So it can go to your brain, it can get processed, and the reflexes get activated in there and then comes back. But when the information is coming back or the signal is coming back, that's an efferent signal, meaning that it's coming from the CNS and it's gonna have a motor function. So it will go to the effector structures like the muscles on your hand, and immediately when this process happens, your reflexes will act and you will take your hand away from the burn so you won't damage yourself. So the muscle is one of the effector structures that could be possible. There could be like glands or many other things as well, which they receive the motor action signal from the CNS and they need to do some action in that area. So these are some general info you need to know. And now we go to dive into the definition and what each of these two important cells do in our body. So first one, neuron. What's a neuron? Neuron is the functional unit of central nervous system. And what do they do? Basically, they are active in protein production. That means that they have a lot of ribosomes in themselves. A lot of ribosomes means that there is polyribosomes in that area. And polyribosomes is usually close to ER, endoplasmic reticulum. So a lot of ribosomes, a lot of ER at the same time, which is nasal body. And the nasal body can be stained. If you guys have already listened to our other videos, they will be stained easily by nasal staining. So, nasal staining. I need to repeat myself for that. Anyway, nasal staining, easily you can see them, and it will be a very good slide for you guys to see. I think it should be on the next or chapter or on the one after that you need to look at the slides from the histology. But here I just explain a general info about them. Now, neurons as well, they get classified in so many different ways. But one classification is the number of processes. And based on number of processes, they will be classified into four cell bodies or four groups. One will be unipolar, then we will have bipolar, then pseudo-unipolar and multipolar. Unipolar, that means that they do not have, if you can see the picture here, they do not have any axons, they only have dendrites. And they're very rare to find out. They're one of the first cells to be produced and their function is very minimal. But a very important example of them is the amacrine cells on your retina or in your retina. Then we will have bipolar cells. The bipolar cells, it simply means that there is one axon here and one dendrite. And where we can find them? Basically, these bipolar neurons, they are special sensory. Their function is for special sensory, and therefore for retina, so the special sensory in that area will be, to look, and for spiral cochlear ganglion, so for hearing. And then, yeah, so that's bipolar. They're going to connect the cochlea to the nuclei in that area. And then there will be pseudo-unipolar. Pseudo-unipolar is very important because they are general sensory. What does that mean? Basically, you will have spinal ganglia anywhere in your body, and especially dorsal root ganglia as well. So these two are in that area, and there is a collection of pseudo-unipolar neurons in them, which allow for their function, general sensory function, to be carried out. And a very good example could also be like the satellite cells or the cranial ganglia cells. And obviously, I already mentioned the dorsal root ganglia. So that's for pseudo-unipolar. They look like a T shape. As if you can see, there is one um, axon and one dendrite, but they're very close to each other, and the cell body is quite far from these two. So it looks like a T shape. That's why we call it pseudo-unipolar. And there will be a multipolar. That means that there is one axon and so many dendrites. And the multipolar divides into three types. There will be a stellate one, there will be a pyramidal one, and there will be a Purkinje one. Now, the stellate one, you can find it in the sympathetic ganglions all around close to your spinal cord. And for pyramidal cells, you can find them in your cerebral cortex. And for the Purkinje cells, which is very important for our faculty, because the founder was part of our faculty, it's found in your cerebellum. So in the cerebral cortex. And multipolar is the most common type of a neuron that we can have, and it's a motor neuron. So it's an efferent neuron, okay? So efferent signals go through them. Now, we go to glial cells. Glial cells, as we mentioned, they are supporting cells, meaning that these ones, they're going to support the neurons in the terms of providing nutrition, insulation, protection. However, unlike neurons, they're not a conducting cells, so they're non-conducting. Now, what's the origin? The origin, all of them will come from the neuroectoderm, with the exception of one, and that is called the microglia. Microglia is another name for the macrophages in our nervous system. They do exactly the job of a macrophage, but they're called microglia. It's good for you guys to know. And as we all know, the origin of macrophages are mesenchymal cells. Okay? Now, let's dive into CNS and PNS. CNS and PNS have different glial cells, which CNS has 4 and PNS has 2. The 4 in CNS are astrocytes, oligodendrocytes. There will be microglia, we already talked about it, and then there will be ependymal cells. Now, the astrocytes are kind of the important one cell in that area. They support and modulate the activity of the neurons in that area. They are star-shaped with different processes, as you guys can see here in red. And there will be two types of them as well. There will be protoplasmic and there will be fibrous astrocytes. Protoplasmic, they're found on the gray matter, meaning the cell body of neurons in that area. And the fibrous, they're found in white matter, meaning the axons of the neurons in that area. And then we have oligodendrocytes. Oligodendrocytes are also very important because they're responsible for production of myelin sheaths around your axon to facilitate and sort of speed up the action potential transmission throughout the neuron. And the difference between the oligodendrocytes and another cell, which is called Schwann cells in PNS, is that both of them, they produce myelin sheath, don't get me wrong. But the main difference is that in oligodendrocyte, they can do that for more than one axon at the same time. So they can produce myelin sheath for more than one axon at the same time. While in PNS, the Schwann cells can only produce myelin sheath for one axon at a time. It's very important for you guys to differentiate this. It could be a very easy question in your final exam, and you can easily get the point because of this. Now, we come to the ependymal cells. Ependymal cells, guys, they basically line the cavities of the CNS in the spinal cord, and they look very similar to epithelial cells, but they're not epithelial cells. And they're part of the choroid plexus. meaning the structure that produce our CSF. And you need to remember that microglia is both present in both CNS and PNS. And so the last sort of cell we need to know about is in PNS, and that's going to be the satellite cells. Now, the satellite cells, they're basically enveloped around the cell bodies or pericardia. Remember this name very well, guys, because it does come quite a lot in these tests, and you will look at it and you're like, okay, what does this mean? It's another name for cell bodies, but it's very tricky in exam. Anyway, so the satellite cells, they envelop cell bodies of the neurons in the ganglia and the ganglia of PNS, obviously. And that is literally it. This is a picture of your cerebellum. And as you guys can see in this area, in the gray matter, if you zoom in, you can see your very nice Purkinje cells. And that's easily, if you see any picture like this on the slide, it will only and only be cerebellum. But in order to convince the professors, you need to zoom in, show them these Purkinje cells on the molecular layer, and then you should be fine. You'll easily get the point, and you can get the credit for that exam. So that is the end of our description. I hope you guys learned this pretty well, and if you had any question, this is my email. I'll be happy to help you. See you guys soon.